So welcome everyone, bonsoir, welcome back to this ongoing series. Still hoping to be live one day, but thanks for coming back. So just to tell you, the EUR G.E.N.E, I say the same thing every time, but I really mean it. The EUR G.E.N.E is here for you. So EUR G.E.N.E has been sponsoring this seminar series, but also lots of other workshops. For example, tomorrow night we have an after work about careers in science communication. Again, it's online, it's free, and I think it's in English. So really, almost every week we're having 1 or 2 workshops. So please don't hesitate to sign up and come. And if you have any ideas of things that we're not doing enough for you, just let EUR G.E.N.E know. So tonight, Manon is gonna be our chair. So over to you, Manon.
Thank you, Jonathan, and thank you everyone for being with us tonight for this 27th seminar. 27. Yes. So tonight we are glad to welcome Dr. Aleksandra Deczkowska from the Institut Pasteur. She did her PhD at the Weizmann Institute of Science on Neuroimmunology in Michael Schwartz Laboratory. Then she moved to Ido Hamid Lab for her postdoctoral to work on immune genomics. Thereafter, she came to Paris to be principal investigator at the Institut Pasteur. She worked where she studied the interactions between immune cells and brain. Tonight, we are pleased to hear her talk about immunological mechanisms of brain aging and neurodegeneration. So thank you, Dr. Deczkowska, for accepting the invitation, and the floor is yours.
Thank you very, very much. So let's get right into this. What we are studying, what's our big question and big problem to solve is brain aging. And this is because brain aging predisposes you for dementia and Alzheimer's disease. And the problem is big because as we are aging as a society, and dementia is a really serious problem. These diseases, predominantly Alzheimer, are incurable, they're chronic, and there is seriously no treatment, and they're fatal. And so it's a big strain on the patients, on their families, and on the medical system. And so we are trying to prevent them. Of course, there are many approaches. People are trying to cure Alzheimer, sure. But some of them already gave up on it. We really, really tried many obvious things that were to do, and everything fails in clinical trials. So another way to do it is to try to stop brain aging. And this is where we are with our research. And this is one problem that we have to deal with. And another one is that brain is hard to cure in general. Brain is isolated in the body. It's not only about neurodegeneration, it's also things like schizophrenia, any mental disease, anxiety. It is hard to cure. Why? One reason is that brain is really isolated in the body. It's really separated from everything else. And even if you have a great drug, especially now biologicals, antibodies, you have to get it through the famous blood-brain barrier. And so it really hits the neurons, and this is not easy to do. And the flip side of this is the fact that the brain is isolated for a reason. It is very fragile. The entire function of the brain, all your memories and abilities are encoded in the structure of the brain, in the architecture of how the neurons are connected. And if your drug gets to the brain, passes the blood-brain barrier, hits the neurons, and does something wrong, the neurons die and there's no coming back. And so people are trying to find other ways to cure the brain. And this is especially in the aging and rejuvenation field. We are trying to figure out what are the interventions, what can we do to modulate maybe the body. And so this would slowly and physiologically modulate the brain. And in the aging rejuvenation field, these things actually work. Things like exercise or caloric restriction or intermittent fasting or parabiosis. So exercise and caloric restriction has been also checked in humans. It's very helpful, at least in keeping brain healthy. And parabiosis has been done in mice. It's an experiment where you take 2 mice and you literally suture their skin together, so their blood is constantly mixing. And so when you expose old brain in an old mouse to young blood for a long time, it gets rejuvenated. And with this idea of trying to cure the brain or prevent brain aging, rejuvenate the brain from the outside, let's go into the topic of neuroimmunology. You can guess that it is an interaction between the nervous system, the brain, and the immune system. But the field is a bit conflicted, honestly, and very confusing. So I decided to prepare a little intro on both immune system, nervous system, and both of them together, and what is the problem of the field, really. And there's many. Again, if it's too simple, I apologize, but I think it's a good way to put everybody on the same page. So 3 minutes course on immunology. Let's go. Immune system is there for you to protect you from pathogens, from damage, and these functions are mostly regulated by immune cells. We have 2 types of immune cells. There's innate immune cells and adaptive immune cells. Innate immune cells are mostly in the organs. Basically, they're all born in the bone marrow, in our long bones and flat bones. They are getting out to the blood, and then they're infiltrating the organs. They're going through the organs, scanning what's going on there. And then they get out to the lymphatic vessel and to the lymph node, which is kind of like a control center of the immune system. And so if the immune cells are scanning through the organ and find something dangerous, like a bacteria, bring it to the lymph node, this is what will start the immune response. And another 1 minute on the immune response. Normally, tissue, again, is scanned by immune cells that are there at all times. They're constantly circulating, checking everything is fine. But let's say it's not fine. Let's say I scratched my skin while gardening. So first of all, I have some cells damaged and dying, but I also have bacteria, probably tetanus, viruses, fungal toxins, and literally dirt in my wound. And so this will start immune reaction. These immune cells here will see that something is wrong, go to the lymph node, call on some help from other immune cells and start inflammatory response. So what I will see is redness, swelling, the tissue will be hot. There's a lot of stuff going on there. And the most importantly is that these cells here are trying to get rid of the damaged cells, the bacteria, the fungal toxin. They're trying to eat it up through phagocytosis and get rid of it. And after they cleared everything, we have a step called resolution, where the inflammation calms itself down, shuts itself down. At this stage, usually the scar tissue shows up, and after a while, we'll have repair. And it will take a while, but I will have a normal functional tissue in this spot after all. So now, this is super important, not only in case you have a scratched skin, but also in regular maintenance of tissues. And this is because you can think about tissue function and tissue aging like about having a bicycle. So let's say you have a new bicycle and it's amazing and it's functional, and it will get old with time and it will function worse than it does. But you can partially prevent it by keeping it cleaned and oiled. And normal tissue functions in a similar way. It functions pretty well, but sure, with time, it will get dirty, it will produce waste, it will excrete some protein aggregates, there will be some leftovers, there will be some dead cells or senescent cells. And the immune system, because it has this ability of phagocytosis, which is pretty efficient, immune system is really helpful to get rid of this stuff and keep the tissue clean and functional. So this was 3 minutes immunology. Now 5 minutes on brain function. And I need to tell you this because we use brain function as a readout. So we rather modulate the immune system, look at the brain about how it feels, whether it's young or old. So I think it's interesting to show you how we measure whether the brain is young or old. And there's a few things we can measure. One is neurogenesis, especially in the hippocampus. It's a brain area that is responsible for learning and memory. This is also abilities that go down with aging. So neurogenesis we measure by, in the hippocampus, counting how many newborn neurons we have. And here they're stained in brown. They're marked with double cortin marker. You can see how many we have in young mice, and in old mice we literally have 2. The drop is 50-fold in mice in aging. We also can, in the same way, stain for BRDU incorporation, which is another marker of cell proliferation. And this works very well in mice. The topic of neurogenesis in humans is very controversial, but in mice, this is what we can do. Another thing that is changing dramatically with aging is the levels of inflammation. I told you that the immune system is very important for maintenance. And if there is too much damage, for example, when you scratch your skin, it will overreact. But with aging, as this waste and protein aggregates and damage accumulates, we may have this reaction as well. The inflammation will start from within. And this is something we can measure. So in the brain, you can measure it by staining for astrocyte marker GFAP and EBA1 on immune cells called microglia. And this tool, you'll have upregulation of the marker, so we can see the stronger staining in aged mice, but we also observe changes in morphology. In young mice, these cells have very long processes and branched and thin. In aging, the cells are partially activated. The processes are shorter, less branched, thicker, and the cell is more round, primed, and this is how they respond to inflammation. This is how they are also prepared to phagocytose more. And finally, the most important brain function that we measure is the actual brain function. Who cares about proteins or how the cells look? We want to see things like cognition, learning, or memory. So in mice, this is actually very simple to quantify. And we go by spatial learning and memory. And this can be, you can think about it like, I parked my car somewhere in Paris, and I'm trying to learn where I parked it so I can go about my business, come back and remember where I left it. So in Paris, I would look for things like Eiffel Tower or metro station or pharmacy, because you guys have a lot of them, to orient myself in space. And so then I can come back to my car easily. We do the same to mice, more or less. So there, I'll talk to you about 2 tests. One is novel location, or novel object location, novel location recognition, where we put mice in an arena. It's a fancy word for a plastic box. On the walls of the box, we put visual cues, the Eiffel Tower, the pharmacy, and the metro station. And in the box, there are 2 objects. Mice are very, very curious about things, and they like to explore. So mice will explore these 2 objects for a while. Then we put them for 24 hours to the home cage. They do whatever mice do in their home cage. And then after 24 hours, we put them back into the arena, but one of the objects is in a new location. What will happen is that on day 1, we will measure for how long they explore both objects. And it will be 50% of the time of exploration per object. On day 2, if mice are fine, they will actually spend way more time with the object that has been displaced. And this is because they're curious. This thing is already part of the landscape, but this has moved. This is interesting. Unless, of course, the mice have impaired cognition. And this is when they, again, spend 50% of the time with which object, like everything is new to them. They don't remember anything. So this we can really, this exploration time, we can put on a graph. And this is what you see for normal mice, 50% on day 1, around 90–95% on day 2. For impaired cognition, we see no change, 50–50 on both days. Another test that we can do is water maze. There are different types of mazes. This is radial arm, the arena, the box. It looks like that. And it's filled with water. Mice hate water, and we make it cold and unpleasant, so they don't want to be there. And this makes them swim also. They can swim, but they really hate it. The only way for the mouse to get out of it is getting to the platform. The platform is submerged though. They cannot see it. They need to learn how to find it. This is how it looks in real life. It's really a pool in which we put water. And the platform is in one of the corners. On day one, mice will take forever to find it. They explore every arm and they're trying to figure out what's going on in general. But after a few trials, they actually remember where the platform is and go there right away. So we can measure both a number of errors and we can measure time. We can put it on a graph and it looks like a learning curve. The steeper, the better. So here, black mice learned, black tagged mice learned faster than the red marked mice. Okay, so now let's go to neuroimmunology. It is going to be confusing because it is confusing still. On one hand, we know perfectly well that immune system and nervous system are connected, both ways. When we have any sort of inflammation, COVID, regular cold, your leg is broken, whatever, you feel like staying in bed, sleeping, and being just on your own. You don't want to interact with people. So here is how inflammation regulates our brain. And it works the other way as well. If I have a headache, which I can take down by taking an anti-inflammatory pill, even if this pill is a sugar, but I believe that it's an anti-inflammatory pill, it will actually cure my headache. This is the placebo effect. So the way of our thinking, our brain can affect inflammatory processes. So we know that, but figuring out the mechanism of that, again, very confusing. And some of it is because there are some things that we figured out 100 years ago that really stuck with us, and we couldn't get rid of these old dogmas that we created 100 years ago. So for example, in 1927, there was a paper in G.E.N.E where they transplanted mouse tissues into rat brain. It was tumors, I think. Yes. So they took mouse tumors, put it into the rat brain. Normally, when you take tissue from one animal and put it into another animal, the immune reaction is very intense. It's crazy. It's a foreign body full of biological material that the body of the acceptor would reject. So in brain, it's not exactly the case. And this is, by the way, how you collect data in 1927. So when you take tumor and put it into the brain, in most rats, it will grow. Everything is fine. Here they put the tumor all together with spleen graft. So they supplemented the tumor with immune cells, and then the tumor gets rejected. So this means that by itself, the brain is kind of poor in immune reactivity. It needs help in order to do something that normally body would just do. And some immune reactions in the brain are very limited. This is further supported by other findings. One was about the blood-brain barrier that we already talked about. Paul Ehrlich injected dye into animals, into the veins, and looked how the dye perfuses the body. And when you inject the blue dye into the mouse veins, you get blue liver, blue lungs, everything is blue except of the brain. Brain doesn't get blue. Brain is clear. The conclusion from here was there is no communication between the bloodstream and the brain. Another finding comes from Pio de Liro Ortega about microglia. So I told you that every organ, in every organ, the immune cells come, survey, get out to the lymphatics, In the brain, it's not exactly the case. The brain only has one immune cell type, and these are the microglia. Only one. Nothing else is supposed to be allowed there. And finally, I told you that from every organ, the immune cells drain, they get out to the lymphatics and go to the lymph nodes, to the immune system control center. In the brain, we were sure that there is no lymphatic for the longest time. In addition, in the neuroimmunology field, people were very intensely studying one disease, and this is multiple sclerosis. It's a horrible disease that compared to other diseases in neuroimmunology, we know a lot about it. It's still pretty horrible. There are treatments that are disease modifying. There's no cure. it is autoimmune. So this is where the immune cells come into the brain to kill oligodendrocytes, and this is how neurons slowly lose function, die, and it ends up in paralysis. But people, you can see a lot of immune activity here inside the brain. And based on this, people generalized it into any immune activity, any infiltration of immune cells to the brain is bad, is detrimental, it's pathology. And so overall, the concept was the brain is immune privileged, an immune system that is outside of the brain has nothing to do with it, only microglia allowed, there is no lymphatic connection, immune and brain should not be mixed. But since then, we are, I think, really the last 15 years only, we are getting a higher resolution here and we are understanding this communication better. So for example, with time we learned that microglia are really not that good at being immune cells. They are the only immune cell type in the brain. But when there is a virus or bacteria, they're really just freaking out and not really protecting like they should. Instead, they're very good at maintenance. They're very good at eating dead cells, getting rid of excess neurotransmitters, pruning the synapses that are not necessary. So they're really kind of gardeners. They're keeping the place clean, but in case of alert, they don't really know how to react. But we also learned more about what's going on in terms of connection of the outside, the periphery with the brain. So for example, in Michal Schwarz's lab in 2007, they found that CD4 positive T cells that are never found inside the brain are actually necessary for learning and for neurogenesis in normal mice. We later learned from Kipnis' lab that the cytokines, these are the signaling molecules that immune cells secrete. Cytokines are very important for regulation of behavior, and it's very complex things like learning, memory, but also social activity or anxiety and kind of bravery of mice or courageousness. Finally, we also found that brain has a lymphatic, and it's right here. It's in the middle of the skull. Just we were sure that there is none, so we didn't really look for it, and suddenly it's there. It's also there in humans. We have also evidence from humans. And finally, most recently, we also found that we need the brain for proper immune activity as well. And this is a paper about immune memory, and you can maybe we can talk about it in terms of COVID vaccine. You're vaccinated. Some of you probably post-vaccination, it was a difficult day. And now when your body interacts with real COVID, it will freak out and reject it very fast because it has already trained to do it with the vaccine. So apparently there's this retrieval of the memory in the immune system is actually controlled by the brain, by neurons in the insular cortex. And now, still, this is super fun, but not everything is clear about how it exactly works. For example, the CD4 positive T cells producing the cytokines, there is no immune cells in the brain. There is only microglia. So where are these T cells? What's going on? They're not in the brain, they're somewhere else. And how do they, from this somewhere else, affect the brain? And this is what motivated scientists in neuroimmunology to really focus on the brain borders. We talked about the blood-brain barrier. This is this very strong sealed vessel that doesn't let anything pass, even the blue dye in ancient experiments. But we also have other places that can be considered connections between the immune system and the brain. This is meninges, the space between the brain surface and the skull, and the choroid plexus, which is located in the brain ventricles. Ventricles are the empty spaces that are filled with cerebrospinal fluids, which we'll talk about more in a minute. And here is where the choroid plexus is located. And this is the blood cerebrospinal fluid barrier, the choroid plexus. The main function of the choroid plexus, this is how it more or less looks, is production of the cerebrospinal fluid. The fluid is very important because on one hand, it's a filtrate of the blood, which is enriched extra with neurotrophic factors. but also things that get transported from the blood, including glucose, vitamin C, thyroid hormone, things that are absolutely necessary for the brain to work. So on one hand, the CSF brings all these things to the brain, and on the other, it flushes the brain from all the waste that the brain produces, all the protein aggregates, including amyloid beta, thing that is really the core of the problem in Alzheimer's disease. And so this is the major function of the choroid plexus, epithelium. This is the key element here. But on the other side, this choroid plexus is very, very sensitive to immune stimuli, and the immune cells are present at the choroid plexus, and different ones, including CD4 positive T cells. There is no microglia here. And this is something that we are looking at in the context of aging. Again, why do we think it's relevant? Because immune system is so good at maintenance, and aging can be considered as loss of maintenance. You don't take care of your bike will break sooner. And these maintenance mechanisms, we think with age go down, they wear off, and we have accumulation of something we can call wear and tear. Just regular damage that happens all the time. Just this time we have a lot of it. Specifically, choroid plexus is great at it because it produces CSF that again flushes all this waste and leftovers from the brain while giving it neurotrophic factors, sugar, vitamin C, and all the goodies. And on the other hand, we have microglia, which I said are very important for maintenance by pruning synapses, also producing neurotrophic factors and phagocytosing, again, all the dead cells and protein aggregates. So with age, indeed, many of these mechanisms are going down. And we think maybe if we can bring them back, if we can fix it, if we can make it work, this would rejuvenate the brain. So now let's go more specifically to what we've done in the past and where now our new fresh lab at Pasteur is going. And I will talk about two things, the choroid plexus regulation and microglia. The choroid plexus, again, it's very important in production of the CSF, key for maintenance of the brain. On the other hand, it's very sensitive to peripheral signals, including immune cells. And because of this being in the middle, we thought in the past that maybe brain ages because of the choroid plexus dysfunction, but the choroid plexus dysfunction is induced from the outside by something else happening in the old body. And in order to study this, we did a pretty insane experiment. We took choroid plexus from young and old mice, but we also took the brain, and we also took many other organs from the body. I think it was 17 and 11 made it to the final paper, and we sequenced everything for RNA to see general signatures, again, hoping that the CP reflects something from the outside and projects it on the brain. Of course, it didn't happen. What we in fact saw is that all organs have a similar signature of inflammaging. This is again because of the accumulation of damage with time. Finally, this maintenance mechanism of the immune system switches to inflammatory response. And so we have the immune response. And the choroid plexus, instead of reflecting anything, gave us a super unique signature of type 1 interferon. Type 1 interferon is antiviral. And so first of all, we freaked out thinking that our old mice, something is wrong with them, they have a virus. Turned out not the case because we took mice from 3 different locations, I think, in the world, making sure that there is no virus. It's very consistent if you go from one animal house to another. And finally, we got human tissues and we observed the same phenomena. We have both the key transcription factor and the secreted factor that maybe is secreted to the CSF expressed at the choroid plexus in old humans. We finally, I skipped the part. We looked at, does it do anything? So we have interferon type 1 and the choroid plexus is very unique, very fun. Does it matter in aging? What does it mean for the brain? And in order to study this, we wanted to block it in old mice and see what happens. But before we get there, we need to identify which mice have the cognitive ability still retained and which ones lost it. I told you that mice lose it, but not all of them. So first we took a really big cohort of old mice and we ran the novel location test. We identified which mice still retain cognitive function and which lost it. Then we only took those that lost it and divided it into two groups. The ones that received interferon beta blocker and the ones that received the control antibody. This was injected into the cerebrospinal fluid. And in this experiment, we tested them again for novel object location. And we found that the ones that received the blocker actually were able to remember the displaced object location. And so at least this hallmark of brain aging we were able to rejuvenate. We also saw increase in neurogenesis. This is the double cortin staining and BRDU incorporation. We also saw an increase in IL-10 production, which is an anti-inflammatory factor. And we observed lower levels of inflammation. This is the young, control old, and this is treated with interferon type 1 receptor blocker. And here we have the GFAP astrocytes and EPA1 microglia intensity. So now the next question was, of course, how does this work? Interferon beta has negative effect on the brain. We can reverse it in aging. Okay, how? What's going on? What's the signaling? Now, the first bet was that it's going through microglia because they're immune cells and they're, of course, at least in theory, very, very sensitive to immune stimuli. And antiviral signaling that spreads to the body should really shine up all the immune cells within reach, alerting them that something is going on. So microglia are very sensitive to that. Many other things that can be present in the brain when there's any sort of problem. And if you take microglia from old mice, in fact, you do see shadow of interferon type 1 signaling upregulated in aging. These are all the genes that fall under interferon type 1 regulation. And we wanted to study the involvement of microglia in the interferon type 1 effect on the brain, but it's very hard in aging. It's hard to keep mice for 2 years if your PhD only takes 3.5. And it is dirty. There's so many things happening in an old body. It's really hard to isolate specific effects of a single molecule interferon beta on your system. So we needed a better model. And we figured out how to manipulate the choroid plexus in a very specific way. We found that one of the serotypes of adeno-associated viruses, if you inject it into the ventricles, into the spaces filled with fluid in the brain, they go very specifically and very efficiently into the choroid plexus epithelium. And they stay there. The virus is not integrating, but the epithelium does not proliferate. And so you can see here the GFP signal, even 10 months after the injection. So having this tool, we overexpressed interferon beta in the brain of young mice, and we tested their cognitive ability. And I apologize, but this is not true. We didn't wait 4 weeks. Maybe the first time around, we actually waited 10 months. Young mice, even with tons of interferon beta expressed from the CP, were fine. We had to wait longer to see an effect on cognitive ability. So this is middle-aged mice, let's say 45 years old. And here we see an effect in black mice in control. You see in the test they're doing better. And then they're doing more than 50%. For the red group, interferon beta overexpressing is 50-50. And this is the radial arm water maze, where the learning curve is steeper for control mice than for the mice that were exposed to interferon for a long time. So using this model, we decided to check, are microglia really involved? And for this, we again overexpressed interferon beta, but this time in mice where some of them did not have interferon type 1 receptor, IFN-I, on their microglia. This is a genetic model. So we had this, we had control, and we had interferon overexpression or control. This is normal mice, normal microglia, control AAV. They are great at identifying novel location. This is what happens when we inject interferon beta and overexpress it. And this is what happens when we overexpress interferon in mice where microglia cannot sense it. The effect is really rescued. And so the whole thing seems to be going through microglia. How? What does exactly interferon is doing to microglia to have this effect on cognition? And we figure out there's several mechanisms that are going on. Some molecules that were linked to excessive pruning of synapses, which means that perfectly fine synapses are just plucked out by microglia and eaten up. This is literally your memories disappearing. We also found other signs of pro-inflammatory activation, including disconnection of microglia from the neurons, which is supposed to be very, very important for keeping microglia healthy and performing normally their maintenance functions. And we also found that some of these effects were regulated by a transcription factor MEF2C, which is downregulated by interferon type 1. And this led us to broader thinking about microglia and microglial regulation in tissue, because on one hand, microglia are again very sensitive to immune stimuli. On the other, if they overreact, if they react like normal immune cells, they will start up this cycle in calling other cells in, mayhem, basically destroying and killing and eating up all the cells that may be damaged and not exactly already dead, but a little underperforming. And there is and the brain again is very fragile and doesn't regenerate. And so these activities in microglia have to be curbed. And they are by local signals, which we called immune checkpoints. So microglia do not react as a normal immune cell would. They react much less. And this is because they're constantly exposed to signals like neurotransmitters. Some of the neurotrophic factors induce this effect. Some of the, again, direct connection with neurons and astrocytes. And it's also intracellularly mediated or driven by chromatin modifiers and by transcription factors. So in this story about choroid plexus, we talked about how interferon beta is upregulated in aging at the site, how it negatively affects cognitive function, and how it all seems to be reflected and mediated by microglia. And in the lab currently, we are further looking at this flow of signal from the outside to the inside in aging, but not only, also in other contexts of physiology and disease. And now our tools are a little bit more advanced. We can use single cell genomics. We are also extensively using the AAVs that I told you about to induce modifications in the choroid plexus epithelium, so here specifically. And we are using all the readouts that I talked to you about. So to summarize this part, choroid plexus, very important for brain maintenance, but also very sensitive to immune stimuli. In aging, we observe dysregulation of this site. And it is interesting to consider that maybe influencing the choroid plexus function from the outside, from the blood side, from the immune system, can lead to rejuvenation of the brain. Okay, now very quickly we go to the second part of microglia and their role in neurodegeneration. Let's start from the problem of Alzheimer's disease. Alzheimer is this horrible, uncurable, fatal, and chronic disease where we keep forgetting things. And this leads to finally functional loss and death. Molecular hallmarks are accumulation of amyloid beta plaques. This is a protein aggregate. Extracellular, we also have tau tangles, intracellular inclusions of protein aggregates inside the neurons. This all causes neuronal death. Now, the immune system over the years has been implicated in this. People kind of felt under the skin that this must be related to inflammation. We have accumulation of waste, also related to aging, but we couldn't figure out what it is. We didn't know which cells are involved. We didn't know what pathways are involved and how is it good these changes that we observe, or are they bad? And finally, some answers came with the single cell technology. So single cell sequencing of RNA gives you the answer about the cell identity without any assumptions. You don't have to guess if it's a microglia or not. You just sort cells and you kind of see. So Hadassky and Shao in Ido's lab did it in a model of Alzheimer's and in wild-type mice, in their aged much later mates. She took a brain together with the borders, and so you see different immune cells here, but still the major population is the microglia. And this is expected, right? We have a lot of microglia inside the brain, some immune cells on the borders. But she also saw something weird. These two subclusters of microglia. Microglia are supposed to be the same all over the place. This is not supposed to be there. You can also see that they're only present in Alzheimer mice and not in the wild type. And so they're unique to the AD model. And because of this, we started calling them disease-associated microglia. When you have a closer look and compare them to regular microglia, which are still present in the brain, even in sick mice, you can see that they have a lot of genes related to phagocytosis, which makes sense. There is protein aggregates that they are supposed to eat, and to lipid metabolism. And it is true that there's a lot of cholesterol and lipids associated with plaques. So it kind of makes sense. Now let's take these super specific markers and try to stain for them in the brain. And in both mice and humans, the cells expressing these markers were found in direct contact with amyloid beta plaques. So these cells are in the heart of the disease. They're really in the middle of where the pathology is going on. Interesting. Now, Hadass also looked at other diseases. She looked at natural aging. She looked at a model of amyotrophic lateral sclerosis. And she looked at other group of Oleg Budowski looked at EE, which is this model of MS, which we already talked about. In all these models, we and others found some small population of DAMs. It's not as extensive as in Alzheimer's, but it's there. And we started asking, what is inducing this phenotype? And our thinking is that everywhere else in the body, bacteria, viruses, or apoptotic cells, any kind of damage will induce activation of macrophages, which microglia are, in fact. And microglia, they are special macrophages. They're isolated in the brain, have all these checkpoints on them, but they still react to neurodegeneration associated signals that can be coming. They can be different in different diseases, but the signature, after all, is always the same. What is interesting about the signature further is that many of the genes that come out as unique or specifically expressed or downregulated in DAM, compared to microglia, they have been associated with Alzheimer's risk. This graph is, I know, complicated to understand, but long story short, you need full ability to produce DAMs to be a little bit more protected from Alzheimer. And if you cannot produce DAMs because you have mutations in some of these genes, your risk of developing Alzheimer's is higher, which in some sense tells you that they're beneficial. Maybe they're protective. They're in a way good. But that's not a real scientific answer, right? In order to approach it scientifically, we need to get rid of them and see if the Alzheimer gets worse or better. And until now, we tried, for example, with TREM2 mice. So TREM2 is this gene associated with Alzheimer's, apparently very important for DAMs. If you knock it out, this is normal mice with normal microglia. In 5 fat mice, this is model of Alzheimer's. They transit to DAM. If you take TREM2 out of the picture, the cells get stuck in the middle. So they didn't transit, but they also didn't quite disappear. They just transformed into something in the middle, which is not exactly the clean model that we need to answer our question of what is their role. We also know that it's TREM2 knockout mice. I told you that the DAM like to cluster around the plaques. They're really at the heart of pathology. In the TREM2 knockout, it's not the case. They do not create this barrier around the plaque. Maybe that's why the plaques get a little bigger. And here the plaques are in red and the microglia are in white. But the real function would be looking at the brain function, at the Morris water maze, at the radial arm water maze, at the location recognition. And in these tests, the contribution of TREM2 to Alzheimer pathology is very controversial. It depends on the model, it depends when you knock it out. And again, like on the last slide, the TREM2 knockout does not mean that you got rid of the DAM cells. So we still don't know really what's going on. We also figured out over the years other modulators of DAM activity, DAM levels, DAM phenotype, but we still do not have a good way to totally kill them. And we are stuck on that. And we're also stuck on the question in the lab about this microglial immune checkpoints. What are exactly the factors that modulate microglial identity, microglial responsiveness to immune stimuli, microglial ability to get rid of waste and keep the brain young? And this is why we are still exploring these questions now in the new lab at Pasteur. Especially we're trying to design new tools to study microglia to approach such questions in a more efficient way. So to summarize the second part, microglia have an important maintenance role, important thing to remember. In Alzheimer and other cases of neurodegeneration, we think that DAM arise. It's a subtype of microglia that has a very special signature. The signature of DAM has been linked to risk of Alzheimer's in human genetics, in a direction suggesting that they can be good, but we still cannot scientifically prove that they are good or bad without good models, and this is something that we are working on currently in the lab. So with this, I would like to end here and acknowledge my lab. This is the people trying to solve these questions right now, with help of our wonderful collaborators and powered by these funding agencies. Little announcement, we joined the global efforts to help researchers coming from Ukraine. I would be really grateful if you know anyone in Ukraine or already escaped looking for a place to stay. We have quite many options at Pasteur for them to host them. And I would like to thank you for your attention, leaving you with this quote, half from Buddha and half from my friend and colleague, Leve, saying that we are shaped by our thoughts, but our thoughts are shaped by microglia. So please remember that neuroimmunology is really what's important. Thank you very much for your attention.
So thank you very much for your speak. It was very interesting. Now it's question time. And I believe there is already some questions. Okay, so to begin with, we have a question from Rosani Singh, who is asking, what does different microglia 1, 2, and 3 represent? Are there specific types of microglia?
Yes, so in this, it has been described in the paper. We have microglia 1 can be considered homeostatic. This is what we have everywhere in a normal, healthy brain. Microglia 3 is the classical DAM, which have the full-blown ability, and microglia 2 are the intermediate kind, and this is where in the TREM2 knockout, microglia get stuck in the middle of this transition from the homeostatic to the disease-associated. This can be, though, very artificial separation because it's a TISNE, it's not biology. We did something with TREM2 knockout, but we don't really know if it's a real trajectory, if the trajectory goes more like to the sides, maybe from the homeostatic. Things to be considered and to be checked. Okay, thank you.
We have some questions from Ranabir Chakraborty, who is asking, does EFN-beta bring about global reactivity of microglia, or is it localized to specific regions?
Yes, so there's a paper scooping us a little bit on that. They took microglia from different regions in aging, and they saw that there is definitely more interferon response in microglia in the hippocampus, which kind of makes sense location-wise because it is closer to lateral ventricle choroid plexus.
We have another question from Hanabi, who is asking, circulation-derived myeloid cells have shown efficient engraftment in the brain parenchyma, albeit the phenotype of these cells are dam-like. Since this approach was able to rescue to a great extent the neurodegenerative phenotype of Gaucher's disease, would you think it would be fruitful to transplant healthy CDMCs and potentially rescue other AD&D phenotypes?
Yes, definitely. There is an entire huge field, which some of us feel is very artificial, working on how do we do engraftment on microglia of monocyte-derived macrophages to the brain to replace the microglia that are, in fact, maybe exhausted in aging, definitely in Alzheimer's. So there is an effort there. My opinion is let's wait and see. Because again, microglia are there cooking in this environment since basically birth. They don't renew from the outside under physiological conditions. Maybe there is a reason for that, and maybe letting macrophages out would be more harmful in the long term. Time will show, but it's a really intense field right now.
Can I ask a question? Can I ask you a question?
Manon is deciding. I don't know.
Manon?
Yes, you may.
Yeah, I just wondered, to what extent can we say that different tissues age at different speeds or at different rates, and the aging of one affects the other? So, for example, if the immune system is aging differently, will that affect the brain? And then will aging of the brain affect the immune system that might then send a message to another tissue? So how do we think about that?
First, we think about how we measure aging. It can be the methylation, the epigenetic signature. There is a scheme for that about how different organs in this sense age at different speeds. And there's definitely connection between these organs and the brain and their dysfunction in the brain. The fact that the brain dysfunction can induce the dysfunction of what's going on outside is rather new, but the opposite would definitely know. Liver dysfunction, any kind of cirrhosis, fatty liver can negatively affect the brain. Any kind of chronic inflammation that can be related to your obesity or diabetes will affect the microglia as well. It's more connected than we thought. And very hard to study.
Okay, so thank you very much again. I think there is no more questions. If people have questions, please ask them already. Otherwise, I'm going to say thank you again, Aleksandra Deczkowska, and thank you everyone for being here tonight with us. Please, you're welcome to come back for the next seminar on the 3rd of May. And we are going to speak about genome instability and carcinogenesis. So that's it for tonight. And welcome everyone again. Thank you. Have a good evening.
Thank you.